Okay, here's another way of looking at the G protein coupled receptor pathways. So what we just did was activate this, separating the alpha from the beta gamma, the alpha having GTP bound on it, such that it can activate adenylyl cyclases and lead to this, the increase in cyclic AMP concentration, ultimately the activation of protein kinase A that controls these things. I mentioned there are many other responses that are associated with G protein coupled receptors, not all of which are activation. So the next one that we can cover is real easy. It's the inactivation of the same processes that we just activated. So I don't know if you can see this all that well, but this says alpha S. The S is for stimulation. So this is a stimulating alpha subunit. This says alpha I. So this is an inhibitory alpha subunit. So even though it's active, GTP bound, its activity is to inhibit things. And among the things that inhibits, and the reason that it got its name, is the decrease in cyclic AMP concentration line right here. Alpha I binds and inhibits adenylyl cyclases, and therefore that's what you get. Never mind these for now. So what else? So those two are both associated with cyclic AMP, making more of it, making less of it ultimately. But alphas do other things too. There's another category called the so-called alpha Q, or alpha Q/11 category of alpha proteins that activate an enzyme called phospholipase C beta, and I'll talk about that specifically in just a second. Right now, just make the connection between alpha Q and the activation of phospholipase C. So this is not going to be dealing with cyclic AMP. Now we're talking about a completely different mechanism. It's going to ultimately lead to a calcium spike by a mechanism that's coming up next. But for now, just make that association. And then there is a fourth, at least, family of alphas, so-called alpha 12-13s, that are involved in things that I'm not going to be talking about. Actually, we'll talk about beta-catenin before too long in a different context. But this is that remodeling of the cytoskeleton that I said I wouldn't talk about, and I'm not gonna. Okay, so before we get down to this business, which is coming up next, I want you to notice this, and that is the beta-gamma subunits of the heterotrimeric G protein now released in the active form from alpha. They do essentially the same thing that the alpha Qs do. They activate phospholipase C beta, exactly the same activity. They do other things too that I'll come back to, but notice that you can get to the same place two different ways with this pathway. So how do you get to that, and what does it accomplish? That's what's coming up next. So in this set of figures, we have figure 1528 in your text, which identifies the catalytic mechanism associated with that activity that was activated by alpha Q and the beta gammas in some cases. So the substrate of phospholipase C is shown here. In this case is PI45 bisphosphate, our old friend PIP2. So in the presence of phospholipase C, as opposed to phospholipase A, B, D, which would do different things, this enzyme catalyzes the breakage of the bond right here to leave you with diacylglycerol. So no phosphates, just the glycerol and the two fatty acids, a very, very, very hydrophobic molecule, and a very, very, very hydrophilic molecule, which is inositol 1,4,5 triphosphate, or IP3. So before I go any further, let me just tell you one thing that you'll maybe appreciate later once we do the PI3 kinase pathway. So this is a molecule that is not PI3. IP3 means something completely different. So the nomenclature here, the PI45, so there's 1, there's 4, and there's 5. So there's the 3 carbon, there's the 2 carbon of inositol. PI3s have a phosphate here. And that, I would say, is maybe the most important phosphorylation reaction in all of biology. Whether there's a phosphate there or not has life or death, literally, consequences. So this molecule does not have a phosphate here, so it is not a PI3. This molecule doesn't either. 1, 4, 5 triphosphate. So there's the 3 carbon, and there's no H there, no phosphate, right? So this is not a PI3 either, but it is an IP3. It's going to be doing something completely different from PI3s, and you say, why do you keep talking about PI3s? Well, we will when we talk about MAP kinase pathways, and it'll be a very important thing. Don't get those confused if you can help it. All right, so but what we have done with phospholipase C beta is produce IP3 and diacylglycerol. It turns out both of those things do something, have downstream functions. And those functions are shown in sort of a schematic form right here. So that reaction right here is shown here as the result of the activated form of phospholipase C. Beta is just one of the isoform families. There are several other Greek letters, and they don't really matter nearly as much as this letter. Excuse me. So here's the diacylglycerol. We're going to use it in just a minute. But first, let's use the IP3. Because it has been clipped from the inositol phospholipid, it's free to go. And it's very, very hydrophilic, so it goes through the cytoplasm, where it encounters a receptor on the surface of the ER. So this is the membrane of the ER, and it has a calcium channel that is activated by IP3. The significance of that is that before IP3 comes along, this channel is closed, and there's a bunch of calcium in here because there are active calcium pumps that make that so. The concentration of calcium out here is always, or almost always, vanishingly low and quite high here and fairly high out here. So there's a process that continuously pumps calcium out and into the lumen of the ER. But now, in the presence of IP3, it serves as a ligand that activates and therefore opens this calcium release channel so that calcium just comes flooding down its concentration gradient into the cytoplasm. So you get these very dramatic spikes of calcium. The classic one is involved in the fertilization process, and this color scheme is calcium being released. So cytoplasmic being the cytoplasm is swept by this wave of calcium over the course of a minute or so, triggered by the opening of this channel. So in the typical cell, what would happen is this calcium does a number of things, some of which, a lot of which aren't shown on here, and one I'll just mention before we go any further, and that is the activation of a protein called calmodulin. We're going to use it later on, but for now, just realize that calmodulin is active in the absence of calcium, but if you add calcium, then it activates it, and that results in activation of other things, in particular calcium calmodulin dependent kinase, so-called CAM kinase, that is not quite as, I would say, biologically significant as protein kinase A, but it's the same kind of idea where you have a protein kinase that can phosphorylate multiple targets and mediate a fairly dramatic response. Another effect of that elevated calcium is activation of a different protein kinase. This one is protein kinase C. Now you can think of the C as being for calcium. So what's happening here is the calcium is binding to a subunit of protein kinase C that was previously inactive and floating around out here in the cytoplasm. So the calcium causes the migration of a still inactive protein kinase C to the membrane, and the significance of being on the membrane is that now protein kinase C can interact with diacylglycerol, the other product of the phospholipase C beta reaction. So once you have calcium and diacylglycerol present in the same location on the membrane, then you have activated protein kinase C, and then downstream processes occur. Okay, so before too long, we're going to get a little bit more specific about this domain, which is actually a subunit of protein kinase C, and I'll just go ahead and give that away. That subunit is referred to as plextrin. I'm hoping at least some of you have heard about plex or pH domains, but we'll come back to those before too long. Okay. So we've now accomplished calcium spikes with G protein coupled receptors. We've accomplished cyclic AMP release and activation of protein kinase A. This is really just the tip of the iceberg. There's all sorts of ways that you can use G protein coupled receptor pathways, all sorts of responses that can be controlled. For instance, there are neurotransmitter receptors that are associated with electrical communication. They're G protein coupled receptors, so-called slow receptors. We'll talk about this a little bit. The visual transduction system uses a G protein coupled receptor. And then there's this. There's a phenomenon associated with vasodilation that I wanted to talk about. Here's your book's version of it. Here is the book that I used last year as a version of it. I like them both for different reasons, although they're showing the same thing. But in any case, when we're talking about vasodilation, we got the blood vessel. So here's the lumen. So as is the case with epithelial cells, lumen is separated from tissue by a single layer of cells. In this case, if it's blood that we're talking about as the lumen, then it'll be an endothelial cell, not epithelial cell. So just imagine the same thing that we've been talking about with epithelial cells, but make it endothelial. So there's the basal lamina, and in this case, there's a layer of smooth muscle that circles the blood vessel that is actually doing the constriction and the dilation business as a muscle contraction event. So that process is, I guess you can imagine, very tightly regulated, and G protein coupled receptors are the key part of that. So if you zoom in on this part of the diagram here, then you're right about here for this diagram. So here's the smooth muscle, here's the endothelial cell, and here is blood, basically the lumen of blood vessels. So that's where it starts with acetylcholine. Once again, we're using it to activate a receptor. In this case, it's another G protein coupled receptor, but in this case, this acetylcholine receptor activates phospholipase C. So you can guess that this is probably going to be an alpha Q pathway, and if it's phospholipase C, then you're going to get a calcium spike. And this, in this instance, that triggers the activation of calcium calmodulin. This time it's not going to be activating calmodulin-dependent kinase. It activates a different enzyme altogether. That enzyme is NO synthase, the enzyme that's responsible for producing the gas, NO, from arginine, the amino acid arginine. NO serves as a second messenger, third messenger, maybe, if you consider calcium the second messenger in signal transduction cascades. And by virtue of being a gas, that means it can essentially go anywhere really fast, but it doesn't go very far. So it's a great way to make short-range, very, very rapid responses, or to control those responses. So here you have it diffusing out of the endothelial cell and into the smooth muscle cell almost as soon as it's made. And it serves as the activating ligand for a soluble receptor now. We don't need to put it on the membrane because NO can just float right through. Doesn't even blink going through the membrane. So the soluble receptor in this case is a cyclic guanylyl cyclase. Sorry, messed that up completely. That enzyme converts GTP into cyclic GMP, the significance of which is that it then goes to activate a protein kinase that is cyclic GMP dependent, not cyclic AMP. That protein is called protein kinase G. Protein kinase G is the multi-substrate protein kinase phosphorylates numerous proteins that are associated with the relaxation of the smooth muscle. Relaxation equals vasodilation. So that process leads to the dilation of blood vessels, and it's the reason that NO is beneficial in preventing heart disease, and it's the reason that nitroglycerin is beneficial for patients with heart disease constrictions in their blood vessels because it leads to the release of NO, which then partially alleviates that constriction. So this process, as you might guess, is regulated. So in the case of cyclic GMP, there is a phosphodiesterase that's specific for it, catalyzing the equivalent reaction of the one that we talked about for cyclic AMP. This one is referred to as PDE5, the fifth member of the phosphodiesterase family, and it converts cyclic GMP into GMP, thus limiting the response. So it essentially reverses the relaxation triggered by the NO, which itself was triggered originally by acetylcholine in the blood. The reason I mention this is because this is the target for a whole bunch of drugs, including Viagra, that are designed to do what they do, that is, prolong the relaxation of smooth muscle cells in the particular area that they target. And the way they work is by inhibiting this, which therefore prevents the breakdown of this, which means that this is active longer, and therefore this is active longer. And that's the way billions of dollars are made.